GRIAS Academy.
100 days, 100 themes, one goal, UPSC Prelims. Hello all, welcome back to the program Mission Surya Phase 3 STEM Revision Program, which we deal with current affairs completely related to science and technology and environment in the Prelims perspective. So let's get started on today's session. So for today our first topic is Cas9 real-time gene editing tracker. We already spoke about Cas9 in the previous class. It is a protein linked to CRISPR to edit the gene for the purpose of gene editing, right? The same process, the same function and all. The only thing is it is engineered to allowing real-time monitoring. Okay, for real-time monitoring, a fluorescent is added to this Cas9. For this Cas9, a fluorescent protein is added on extra, which produces glow. Means the fluorescent light, green fluorescent light will be shown. Okay, see what happened in late 2025. In late 2025, scientists at the Bose Institute, Kolkata, engineered Cas9, a novel CRISPR protein that emits light while performing gene editing, solving a major limitation in genetic engineering by allowing real-time monitoring. Because in gene editing process, by emitting this light, what happens is while this gene editing and all, we can clearly see that yes, we need this specific gene to be edited. So is it targeting the same gene or not? We can monitor in the live. Okay, this is the major limitation in the genetic engineering background. Okay, now so let's see what is Cas9. Cas9 is a bioluminescent variant of the traditional Cas9 enzyme, okay, which is the molecular scissor utilized in the CRISPR technology. Moving forward, let's see the composition. What happened? It is created by fusing the standard Cas9 protein with split nanoluciferase enzyme. This Cas9 protein is now fused with split nanoluciferase enzyme. Okay, from where this nanoluciferase enzyme is derived from? It is derived from deep sea shrimp proteins. Okay. It is derived from deep sea shrimp proteins. The problem it solves is conventional Cas9 is completely invisible. We cannot see where this protein is going and editing the gene. However, coming to the Cas9, it is visible, right? Previously, observing the gene editing process requires fixing or breaking open the cells. Cells has to be open if we wanted to see the gene editing, which destroyed them and made real-time tracking impossible. Okay, now let us see the mechanism. Reconnection. The nanoluciferase enzyme is split into inactive pieces. It's split into inactive pieces when the Cas9 protein folds correctly inside and living cells to execute a target DNA cut. These inactive pieces are brought into close proximity. This is what happens in reconnection. Then bioluminescence. How this bioluminescence is given? The pieces are reassembled. Restoring enzymatic activity and producing a visible light signal. Okay, which glows like a firefly. Everyone knows this firefly, right? So like that firefly, it gives the bioluminescence. Next, which is non-destructive, means this light emission allows scientists to watch the editing process. The gene editing process, scientists can directly see happening in the living cells tissues, and even plant lives without harming the host organism. Without harming the host organism. As like in Cas9, what we have to do? We have to break the cell and see if it is happening or not. But in Cas9, it is not that case. A bioluminescence light is being emitted without harming the host organism. Okay, this is the mechanism. Next, let's see the properties. Cas9 pioneers the emerging scientific field of the Terra tracking. Terra tracking means the visual tracking of molecular gene therapy in motion. Next, thermal stability. It is highly stable and maintains its structure and enzymatic activity at higher temperatures compared to the conventional Cas9. Conventional Cas9, what happens is it is appropriate only for few conditions and few temperatures. But compare this Cas9 can this enzymatic activity at higher temperatures compared to conventional Cas9 it can carry. Its sturdiness is a critical factor for the success of in vivo gene therapy delivery. Next, see, coming to the precision in HDR, it increases the precision of homology directed repair. HDR means homology directed repair. HDR is the specific DNA repair process. It is a specific DNA process essential for fixing hereditary mutations. Hereditary mutations means which pass from generations to generation, from from father to child, from a mother to child, from the parents to grandchildren. This is the hereditary thing. So, it increases the precision of homology directed repair means HDR is this specific DNA repair process essential for fixing hereditary mutations linked to genetic diseases like sickle cell anemia and muscular dystrophy. These are sickle cell anemia and muscular dystrophy are few hereditary diseases which is carried from parents to children. Okay, those also can be treated using this one. Okay, next. Coming to the agricultural applications, because it can be tracked in plant systems also, it paves the way for safe, non-transgenic applications in crop improvement and agriculture biotechnology. In the same way, in agriculture also it is used because when a plant has to be modified, a certain gene has to be regulated. When we use CRISPR-Cas9, what happens? It cuts the gene, everything is fine. But if a scientist wanted to see whether the gene is cutting properly or not, whether it is happening clearly or not, they used to break the cell and see if it is happening or not. So breaking the cell and all, it is something different. Rather, this Cas9, what happens? It behaves in the same way like Cas9 only. The only thing is it produces the bioluminescent light so that the gene tracking, the gene editing process is clearly seen by the scientists. Okay. Next, see the comparative analysis, conventional Cas9 and versus global Cas9. The examiner might trick you, can ask you in the match can ask you the conceptual clarity of Cas9 and Cas9. Let's see few features. Features of conventional CRISPR-Cas9 is it is invisible in living cells. However, this engineered Cas9 emits bioluminescent light to glow. Next, monitoring. It requires fixing or destroying cells to observe. With coming to Cas9, real-time tracking in living tissues. Okay. Next, enzyme composition. What is the enzyme composition of Cas9? It is standard Cas9 endonuclease. Coming to the Cas9, it is fused with deep sea shrimp nanoluciferase enzyme. Okay. Next, the thermal stability. Coming to the thermal stability, these conventional CRISPR-Cas9s are works at standard temperatures only. While coming to engineered Cas9, it is highly stable at elevated temperatures also. Okay, these are few differences which is useful for your UPSC Prelims examination. Next, moving on to the next topic. GLP-1 drugs. It is glucagon-like peptide 1 receptor agonists. Means it is basically used for treating weight loss, type 2 diabetes, basically. Generally it is used in, these drugs are used in this. See, let's see what is it. The Drug Controller General of India, the Drug Controller General of India issued a national advisory in March 2026 to curb the unregulated sale, misleading advertising and unsupervised use of GLP-1 drugs for weight loss. Basically, everyone is going back around with weight loss therapies and all. They are taking so many injections, tablets and all for getting weight loss. So, the Drug Controller General of India issued a national advisory in March 2026 to curb the unregulated sale, misleading advertising and unsupervised use of GLP, means glucagon-like peptide 1 drugs for weight loss. Okay. Next, what is GLP? We'll see. GLP is a naturally occurring hormone in the human body. It is the natural occurring hormone in the human body, which is released in the gut after consuming food in response to rising blood sugar levels. After eating, generally when we take carbs and all, like carbohydrate food and all, generally our body after digestion, it releases the insulin. It is the basic mechanism which happens every day. So this is the hormone. GLP-1 is the hormone in the human body which is released in the gut after consumption of food in response to rising blood sugar levels. That's why sugar patients always ask to have less white rice, what we eat rice, because it produces more insulin in the body because it has a high carbohydrate content. Now, what is GLP-1 drugs? These are hormone-based medications. These are hormone-based medications engineered to mimic the natural GLP-1 hormone and prolong its action in the body. Okay. See, these are hormone-based medications. These GLP-1 drugs are hormone-based medications engineered to mimic the natural GLP-1 hormone in the body and prolong its action in the body. To prolong its action as similar in the body. Okay. Next, let's see what's the mechanism. These drugs operate on a multi-pronged approach to manage metabolic functions. To manage the metabolic functions, these drugs are operating. Like insulin stimulation. Insulin stimulation means they trigger the pancreas. Once we have this GLP-1 drugs and all, what happens? They trigger the pancreas to release insulin. Okay, to release the insulin, moving glucose from the blood into cells. Moving glucose. Insulin means glucose only. Glucose moves into blood. Okay. Next, glucagon suppression. They block the release of glucagon. What is glucagon? Glucagon is a hormone from the liver that increases blood sugar. So that if we take this GLP-1 drug, what happens? It reduces it blocks the release of glucagon enzyme, glucagon hormone, which is released from the liver, that increases the blood sugar. Okay. Next, gastric emptying. These slow down the movement of food. These drugs help in slowing down the movement of food out the stomach, increasing satiety, means the feeling of fullness, and reducing appetite. Generally, this is what a weight loss thing is happening, right? See what happens. This first one, insulin stimulation. After having food only, the GLP hormone gets stimulated, means the insulin gets released. Before taking food only, if we take these drugs and all, insulin is automatically generated in our body and it is going into the cell via blood through cells, so that the cells get oxygen from it. Okay. Next, that is one thing. So we won't feel of having food and all. Next, glucagon suppression. In the sense, what happens when the glucagon hormone is blocked, so that the body doesn't release glucagon hormone. Okay. Next is gastric emptying. Gastric emptying in the sense, they slow down the movement of food out of the stomach, which increasing the satiety, means the feeling of fullness, not having food and all. Okay, and reduces the appetite. It reduces the human's appetite also. This is what the GLP-1 drugs are doing. See, few key variants and administrations. We'll see. These are injectable and oral, right? Available in both forms, both injectable and oral. What are the major drugs? Semaglutide. Semaglutide is one which is most widely known, and liraglutide, tirzepatide, which is newer targets multiple metabolic pathways, dulaglutide and exenatide. These are few drugs, major drugs of GLP-1. Next, see for approved medicinal uses. For this is not completely bad, which we can say. It is useful in few things also. Few are approved also. We'll see what are approved. The primary is treatment of type 2 diabetes. In the sense, blood sugar levels will be controlled. Next, secondary is clinical obesity management. Clinical obesity management, where lifestyle changes fades. Next, the tertiary thing is reducing cardiovascular risks in specific diabetic patients. Why basically the diabetes patients are targeted means, as we see, it blocks the glutathione enzyme also, okay, which is released from the liver, right? Yeah, glucagon enzyme which is released from the liver also, it blocks, and it decreases the glucose levels also. I mean, insulin also is decreased. Okay, next, coming to severe health risks. Organ implant. Risk of pancreatitis, because it is blocking the natural function of pancreas is to release that glucagon hormone, but it is blocking. It may lead to pancreatitis, means inflammation of the pancreas. Next, acute kidney injury also, and severe bowel obstruction, because it is lowering the food, so it may lead to severe bowel obstruction also. Next, hormonally, long-term use carries a rare risk of thyroid-related complications. A rare risk of thyroid-related complications may also happen. Next, what is the regulatory status in India? How these GLP-1 drugs are being regulated in India means Schedule H status. These drugs are given Schedule H status, where these are strictly prescription only drugs. These drugs are given only on prescriptions. Okay, they can only be legally prescribed by qualified specialists. Legally, when a doctor who belongs to endocrinologist, cardiologist, internal medicine, when they write on a prescription to take this GLP-1 drugs, then only the medical practitioner can give the drugs, or else no. Regulatory action. March 2026, DGCI intensified surveillance on online platforms, wellness clinics and pharmacies. Strict penalties, in the sense license cancellations, are enforced again over the counter sales. Without any prescription and all. If the wellness institutes or pharmacies or any online platforms are selling these drugs, their licenses would be cancelled, which is authorized by whom? Which is ordered by DGCI. Okay. Next, moving on. So moving on to our next topic. Our next topic is SHUKR gene and the evolution of flowering plants. Okay, let's see what is this gene is now. Researchers at the CSIR, Center for Cellular and Molecular Biology, CCMB, Hyderabad, they have recently discovered a gene called SHUKR gene. SHUKR gene, which shedding light on the molecular mechanism behind the plant reproduction and evolution. On plant reproduction and evolution. Right? Which gene it is? SHUKR gene, which is identified by, which is discovered by CCMB, Hyderabad. Okay? So, let us see what is this gene is. A newly identified gene found in flowering plants, especially in the angiosperms. Angiosperms are nothing but the plants, the flowering plants, and which have seeds inside the fruit. Basically these are angiosperms. Coming to etymology, why it is named SHUKR? Because the basic meaning of it is sperm or seed in Sanskrit or Hindi, because it is critical role in the development of plant reproductive cells. Okay, next. Let's see the core mechanism and function of this gene. What happens? F-box gene regulation. F-box means, basically, now let's see what is F-box. F-box is a group of proteins. These are group of proteins. The primary function of SHUKR gene is to control a specific group of genes known as F-box genes. These proteins are targeted mainly. Okay? Next, coming to the pollen development, these F-box genes are responsible for managing the proteins required to develop healthy, viable pollen. Okay? What is pollen? Pollen is a male gametophyte of a flowering of a seeded plant. Okay? Next, coming to the evolutionary significance, Charles Darwin, everyone would be knowing about Charles Darwin. Charles Darwin. He is called as father of evolution. Right? Everyone must be knowing about this. Charles Darwin. He is the father of evolution. Charles Darwin once coined the word abominable mystery. It is basically like abominable mystery of angiosperms. Charles Darwin famously referred to the sudden explosive evolutionary appearance, evolutionary appearance and rapid diversification of flowering plants in the fossil records. He mentioned it as abominable mystery. Next, what is the solution for it? The researchers discovered that the SHUKR genes and its associated F-box genes, these evolve exceptionally fast. This rapid molecular evolution provides a genetic explanation for the sudden rise and massive biological success of flowering plants throughout evolutionary history. Okay, just remember, this gene is being identified, was discovered by researchers at CSIR, Center for Cellular and Molecular Biology, CCMB, Hyderabad. Okay, and remember about it is a gene found in flowering plants, and remember about this F-box gene regulation. Few F-box protein genes are being targeted, so remember about it, and the pollen development. Okay, next. Moving on to the next topic. Our next topic is mitochondrial replacement therapy. What is it? Mitochondrial replacement therapy. This is basically in market it is being calling as 3-parent. It is called as 3-parent baby. Why it is called 3-parent baby? Let's look into it. Because see, mitochondrial replacement therapy, commonly referred to as the media as 3-parent baby. Okay, 3-parent baby technology, because it is a highly specialized IVF technique. IVF means in vitro fertilization procedure, which is designed to prevent the passing of incurable mitochondrial diseases from mother to child. Okay, generally a child carries mitochondria of a mother, right? So carries the mitochondria of the child, basically carries the mitochondria of mother. If mother is infected with any diseases, it will be transferred to child through mitochondria only. Okay, now happened in this is, let's see. See here, this is the parent's embryo. Okay, parent embryo with unhealthy mitochondria, and the two nucleuses, one from father and one from mother, is being fused here. So what happened? This is the donor embryo. Donor embryo is taken in this case, like which has healthy mitochondria. Okay. Now the nucleus from the donor embryo will be removed. The nucleus from the donor embryo will be removed, and this parent nucleus is inserted into the donor embryo, leaving the mitochondria, which is unhealthy mitochondria of the parent embryos. This is what happens in the 3-parent baby technology. This is completely a in vitro fertilization technique, IVF technique. Okay? Now, see, what is the science of mitochondria now? Before understanding the therapy, you must know what is mitochondria is. Basically, the mitochondria is known as the powerhouse of the cell. Okay? They generate most of the chemical engineers needed to power the cell's biochemical reactions. Any reactions which has to be carried out in the body, it is carried out by mitochondria. Coming to the genetic independence, mitochondria have their own small pieces of DNA. When we see the structure of mitochondria, it has few structures like this. Sorry, my drawings are too bad. It has structures like this. So, mitochondria has their own pieces of DNA known as mitochondrial DNA, which is separate from the nuclear DNA. Generally, a body consists of nuclear DNA, not the mitochondrial DNA. Mitochondria is present in our body where only it has, independently they have the DNA within it. They are called the powerhouses of the cell. Next, maternal inheritance. What happens? The mitochondrial DNA is inherited exclusively from the mother to the child. Okay? The sperm's mitochondria destroyed during fertilization. When looking into the maternal inheritance of the mitochondrial DNA, we can see the mitochondrial DNA is inherited exclusively from the mother to the children, where the sperm mitochondria, the sperm also has the mitochondria which is destroyed during the fertilization process. Only the mother mitochondria, which has mutated mitochondrial DNA, she will pass the defected to her children. Okay. Next, let's see the mechanism. See, mitochondrial involves replacing the defective mitochondria. It replaces the defected mitochondria in the biological mother's egg with healthy mitochondria from the donor woman. There are two primary techniques used. As I told you, there is the mother's egg with the defected mitochondria, and this is the donor egg with healthy mitochondria, right? Now, replacing the defective mitochondria in the biological mother's egg. These mitochondria are removed from a donor woman. There are two primary techniques are used. What are those two primary techniques? First is the maternal spindle transfer. By maternal spindle transfer means the nuclear DNA is removed from the mother's egg. The nuclear DNA is removed from the mother's egg. It is inserted into a donor egg. Means the nucleus from this mother is removed and inserted into the donor egg by removing the nucleus of donor egg. Okay, which has healthy mitochondria that has had its own nucleus removed. Okay, this reconstructed egg is then fertilized by the father's sperm in the lab. Now, this donor egg with mother's nuclei and the donor mitochondria is fertilized in the IVF process with the father's sperm. This is one method. Now, coming to the second method, that is pronuclear transfer. Pronuclear transfer in the sense, both the mother's egg and the donor's egg are fertilized with father's sperm. Like in this case, what happens? Both the mother's egg and the donor egg. Both are fertilized with the father's sperm. Okay, then before they start dividing, the nucleus from the donor's fertilized egg is discarded. Before fertilization, before the nucleus get fertilized, this nucleus from the donor egg will be removed in the donor mother. Okay, the nucleus from the mother's fertilized egg is then transferred into the empty donor egg containing healthy mitochondria. This is another method, that is pronuclear transfer. One is the maternal spindle transfer. Next, these two methods are used to in mitochondrial transfer to woman. Next, what is this? The three-parent concept mean? The three-parent concept means the resulting child carries genetic material from three distinct individuals. What are they? The biological father, the biological mother and the female donor. The biological father contributes the nuclear DNA via sperm, whereas the biological mother contributes the nuclear DNA via the egg. And the female donor, she contributes the mitochondrial DNA. So, this three-parent child has, carries the genetic material of three individuals: the biological father, the biological mother and the donor mother. Next, what is the purpose and applications of this? Let's see this purpose. The primary goal is to eliminate fatal, incurable mitochondrial diseases such as Leigh syndrome, which affects the central nervous system, which comes from the family lineages, means family heredity. This syndrome will come. Next, it does not cure a person. It does not cure a person which is already infected by the mitochondrial disorder. It only treats preventive reproductive technology. It is used only in the preventive reproductive technology. Okay. This is one. So, moving on to the next topic. Let's see, the next topic of this is Genome India Project and Sequencing Initiatives. What is Genome India Project? Genome India Project is numbers behind the project. We will see what are the numbers behind the project the government has come up with. See, genome sequenced are till now 10,074. Genetic variants which are found are 180 million and completely novel variants are of 7 million. The data generated by this is 8 pp's and institutions involved are nearly 20 institutions are involved, and neighboring global databases are 44 million and plus databases have been there. Okay, so let's see the context of it. The Genome India project have recently been published in the journal Nature Genetics. Nature Genetics, bringing it into the current affairs spotlight. Okay, what is Genome India project? The core details are: it was initiated by Department of Biotechnology. It is initiated by the Department of Biotechnology and involves a large-scale collaboration of 20 scientific institutes across the country. As I said, 20 scientific institutes have been included in it. Okay, what is the primary objective of GIP? To build a comprehensive indigenous catalogue of genetic variants that accurately reflects the unique and vast genetic diversity of Indian population. So, what is the primary objective of Genome India project? Is to build a comprehensive indigenous catalogue of genetic variations that accurately reflects the unique and vast genetic diversity of the Indian population. This is the major primary objective of Genome India project. Next, let's see the data storage of it. Have a note on it, it is highly important for prelims. The IBDAC is all data generated from this project. The data which is completely generated from this project, either it be database, either it be the institutional data, either it be the any project data, it will be stored under the Indian Biological Data Center, IBDC. Where it is located? It is located in Faridabad, Haryana. Okay. Next, coming to the significance, this IDBC, Indian Biological Data Center, holds the distinction of being India's first national life sciences data repository. Okay. This is the first national life science data repository. Next, what is the support of it? It is also supported by the DBT, Department of Biotechnology. Moving forward, see. We'll see few associated genome sequence initiatives in the match the following manner. In UPSC also, in a prelims examination, in match the following manner, these questions can be asked. So, see. What is the initiative? Indian Gene Programme. Indian Gene Programme's nodal agency is CSIR, Council of Scientific and Research Institute. Scientific and Industrial Research. What is the key objective of the Indian Gene Programme? It is mapping genomes of diverse Indian ethnic groups. Next, one day, one genome. This is the nodal agency for one day, one genome is Department of Biotechnology. The key objective of it is highlight unique bacterial species found in India. Next, the Human Genome Project. The Human Genome Project's nodal agency is the international collaboration. Okay, the key objective is the fundamental project aimed at mapping the entire human genome. Next, 1 lakh genomes project. This is where it is in England. England's pioneer initiative to sequence 1 lakh genomes. England has took up this initiative, 1 lakh genomes project, where England initiates to sequence 1 lakh genomes. Next, international HAP map project. It is internationally, the nodal agency is internationally, like analyzing over 1 million variants across African, Asian and European ancestry groups. Okay. Next. These are associated genome sequencing initiatives, few initiatives. So, in UPSC also you might get questions related to this in the match the following pattern. So, have a look on it. Next, moving on to the next topic, India's first gene edited sheep. India's first gene edited sheep, that is Tamim, which is also called as T1. Okay, India's first gene edited sheep. Nice. So, what happened in late 2024, early 2025, scientists at the Sher-e-Kashmir University of Agriculture, Sher-e-Kashmir University of Agriculture, Sciences and Technology in Srinagar successfully developed India's first gene-edited sheep at Kashmir Merino Lamb, a Kashmir Merino Lamb under a project funded by the ICAR, Indian Council of Agricultural Research. Okay. But in the Kashmir University, scientists have developed genetically edited sheep. Okay. Where is this university located in? In Srinagar. Okay, which is funded by which one? It is funded by ICAR, Indian Council of Agricultural Research. Now, coming on to the details, see, you can see here, India's first gene edited sheep. This is. Okay. Next, coming on to the details, the animal. What is the core animal? Core animal is the Kashmir Merino lamb. The Kashmir Merino lamb named Tamim. Tamim means also referred to as T1. Okay. What is the technology which they used? Scientists utilized the Nobel Prize winning CRISPR-Cas9 genetic scissors, a molecular scissor which is used for gene editing to make precise edits at the embryonic stage. At the embryonic stage only, this using this CRISPR-Cas9 technology, they edited the gene for developing this sheep, T1 sheep. The targeted gene. What is the targeted gene in the sheep is myostatin gene. Myostatin gene is targeted. Okay. Next, coming to the mechanism of action, how it works. How it works means, let us see what is the role of myostatin gene. In vertebrates, generally in vertebrates, the myostatin protein acts as a negative regulator of muscle growth. In vertebrates, the myostatin protein acts as a negative regulator of muscle growth. Simply put, its natural job is to it breaks on muscle development, so muscles don't grow indefinitely. This is the basic function of the myostatin gene, myostatin protein. Now what happens? What is the edit? By using the CRISPR-Cas9 technology, researchers knocked out or disabled the specific myostatin gene in the lamb's embryo. In lamb's embryo only they disabled the specific myostatin gene so that the sheep can develop more muscle. Okay, but what is its role? Basically, it brakes on muscle development, so muscle don't grow indefinitely. This is what the basic function of myostatin gene. By using the CRISPR-Cas9 technology, this myostatin gene is knocked out in the embryonic stage itself only. What is the result? Without the gene limiting its growth, the sheep exhibits a expected 30% increase in muscle mass compared to the normal sheep. Okay, this trait naturally exists in European breeds like the Texel sheep, but is absent in indigenous Indian breeds. In Indian breeds, we do not see sheep with this type of muscle and all. It is generally seen in European breeds like Texela sheep. Okay? No. What is the significance for India in food security? It promises higher meat output per animal, addressing rising meat demands without requiring a massive expansion in livestock numbers. Next, resource efficiency. More meat from fewer animals. More meat from fewer animals means less pressure on agriculture land, water resources, and reduced methane emissions per kg of meat produced. Okay, this is what the significance for India with this T1 sheeps. Next, see, few times the UPSC examiner might trick you with these things. Gene editing, concepts like gene editing versus gene modification. See, what is gene editing and what is gene modification? We'll see on this. How is gene editing mechanism is carried out? It makes precise tweaks or deletions within the organisms owing existing DNA. Next, what genetic modification does? It introduces the foreign DNA. It introduces the foreign DNA from completely different species. Foreign DNA is absent in this gene editing process because thymine contains zero foreign genetic material. Example. And in genetically modified, it is present. Foreign DNA is present. Example, BT cotton contains genes from a soil bacteria. Like that, even BT brinjal and all. Next, regulatory burden. What is the regulatory burden on gene editing processes? Generally faces fewer regulatory hurdles globally because the changes could technically occur via natural mutation. It occurs only through natural mutation. But comparatively to coming to gene modification, genetic modification, what happens is faces intense regulatory scrutiny due to ecological and health concerns regulating foreign DNA. By using foreign DNA, many health issues may also occur. That's why it faces few regulations from the ecological and health concerns also. Okay, now this is what examiner might trick you between this gene editing and genetic modification also. So have a look on it. Next. Moving on to the next topic. Our next topic is the global pioneer context, the world's first. We have seen the India's first indigenously produced genetic sheep, gene edited sheep, that is T1. Like globally, if we see, the world successful gene edited sheep utilizing CRISPR-Cas9 technology were developed by researchers in China. Indigenously India and globally it is China. Okay, this university, Nanjing University and Xinjiang Agricultural University, around 2014 and 15, they have developed this gene edited sheep using the same technology, the CRISPR-Cas9. What is the target? Similar to current Indian efforts, the Chinese scientists targeted the same myostatin gene, okay, to create sheep with enhanced muscle for better meat yield. Okay, next, recent global advancements. Recently, what has happened? Recently, global research has moved beyond simple CRISPR cuts to adenine-based editing, a highly precise tool. Rather than the CRISPR tool for editing the gene, they started using adenine-based editing. a highly precise tool that changes a single letter of DNA without breaking the double helical model of double-stranded DNA to target genes like FEC-B, which controls litter size and fecundity. Okay. Everyone knows like double-stranded DNA is made up of amino acids A, T, G, C, again A, T, G, C, and complementary stand for this U, A, C, G and all. This is the complementary strand and this is the double-stranded DNA gene. Okay. So by using adenine base editing, this tool will eliminate the single letter of DNA without breaking the double helical model. This is what the global innovation at present. Okay. Next. So we have seen already India's first is Tamim T1, the animal in late 2024, earlier in 2025, scientists at the Shiri Kashmir University of Agricultural Sciences and Technology developed the first gene edited sheep, a Kashmir merino lamb named Tamim. Okay. Nodal agency, which nodal agency supported the project? The project was funded by the Indian Council of Agricultural Research, ICAR. What is the result of it? By knocking out the myostatin gene, Tamim exhibits are expected 30% increase in the muscle mass, a trait found in European breeds but naturally absent in the Indian breeds. Okay. What is the mechanism of myostatin knockout? It is a protein. Myostatin is a protein that acts as a negative regulator for muscle growth in vertebrates. Its natural biological job is to tell muscles to stop growing. But by knocking out this myostatin gene by the gene editing process using the CRISPR-Cas9 technology, the muscle is built in the sheeps. Okay. See, the editor. Scientists do not add a super muscle gene. Instead, they use CRISPR scissors to cut and disable the myostatin gene without the biological breaks. The animal naturally develops the significant more muscle. It can give the 30% yield, more yield of muscle. Okay. Next, let's see the comparative concept of gene editing versus cloning method. Okay. By cloning, basically we know this, the dolly trap. By cloning, we have developed the dolly. See the features we'll see. The dolly sheep in 1996 it has been developed in UK, and the Tamim of gene edited sheep which is developed in China and India. What is the technology which is used in the dolly sheep in 1996 is the cloning method, means the somatic cells and nuclear transfer. Coming to this Tamim, gene editing, CRISPR-Cas9 base editing technology is used. Coming to the genetic outcome, exact genetic copy, a duplicate of a single adult sheep is produced. That is the dolly sheep. Next, a unique organism with a specific targeted alteration in its DNA is happened. What is the purpose of developing the sheep? Is to improve mammalian reproduction from adult cells. In this, to enhance specific economic traits like muscle mass, disease resistant, wool quality and all. In this Tamim. Okay, Tamim sheep. Next, moving on to the next topic. UNESCO's Global Ethic Framework on Neurotechnology. Now neurotechnology has also come up. Means, see, during its 43rd General Conference in Paris, which is held in November 2025, UNESCO adopted the world's first global normative framework on the ethics of neurotechnology. This responds to the rapid advancements in the brain-computer interferences, AI decoding and consumer neuro devices. Okay. Next, let's see what is neurotechnology. Neurotechnology is the devices, procedures and systems that access, assess or act on neural systems. Basically like our brain. Okay, especially the human brain. Example, brain computer interfaces like Neuralink, deep brain stimulation DBS, for treating Parkinson's disease, memory loss. Okay, and non-invasive tools like consumer earbuds or glasses that track the brain activity and emotional states and eye movements. These all are few examples of neurotechnology. Moving forward, see, let's see the core neuro rights which are established. Okay, recognize and aims to protect emerging neuro rights. What is it? Mental privacy and brain data confidentiality. Means this neural data reveals thoughts, intentions and emotions of a human. So it is very dangerous. Someone can misuse it also. Okay, so that's why few rights are come up. What is it? Mental privacy and brain data confidentiality means the neural data reveals thoughts, intentions and emotions. It is classified as uniquely sensitive personal data and cannot be extracted, commercialized or sold without explicit informed consent. Okay, without any consent also, this information should not be extracted or commercialized. Okay, next, cognitive liberty means the freedom of thought. The absolute right of an individual, to freely choose whether to use or to refuse the neurotechnology. It is completely dependent on the person's individuality, the right to thought, freedom of thought. Next, the psychological continuity means the mental integrity of a person. Protection of an individual's personal identity, algorithms and external neurostimuli must not be allowed to alter a person's core identity, free will or decision making without their consent. By this neurotechnology, everything, whatever we think, whatever our emotions are, whatever the thoughts are, everything are monitored by this neurotechnology process, because it is completely our brain in one's control. Because so, without any consent or anything, nothing have to be done. Neurotechnology process also, without a human consent, it cannot be done. A person's right, freedom, like if they wanted to adopt the technology, they can, or else no. It is completely the person's wish. Okay. Next, let's see few prohibitions on this neurotechnology. Okay. See, the framework explicitly calls on member states to strictly regulate the ban on the use of neural data. Okay. The framework explicitly calls on a member state to strictly regulate or ban the use of a neural data for workplace surveillances. In workplace surveillances like mandating employer or HR neuro screening or using brain data to monitor employee productivity and stress levels. Highly they can use it in the workplace surveillance also. So it is banned, it shouldn't be used. Next is political micro-targeting. In political micro-targeting, what happens? Profiling brain signals to manipulate political choices or voting behavior. This can also happen through neurotechnology. That's why this is banned. Next, commercial manipulation. Commercial manipulations means using neural data in recommender systems like for nudging, addictive tech or manipulative marketing. All these can be done through neural technologies, so these are also banned. Discriminatory profiling. Using brain data driven markers for making decisions regarding health insurance premiums and all. This is also a harmful one. So by all this, neurotechnology is both good and bad, right? Anyone who is handling this neurotechnology, whenever they know and all, they can handle a human brain easily. In the sense, we will become a toy in someone's hand. Okay. Next, India's regulatory contents. What is the India's regulatory content on this neurotechnologies? Currently, India does not have a dedicated law. Doesn't have any dedicated law explicitly addressing neurotechnology or the protection of neuro rights. India doesn't have any dedicated law. So oversight relies on broader frameworks like the ICMR guidelines, the Drug and Cosmetic Rules and the Digital Personal Data Protection Act. These are only few which is present in India. Coming to the global context, Chile is currently the first country to constitutionally protect mental integrity, and California passed specific laws protecting citizens' brain data in 2024. Only these two countries have come up with it. Okay. Next. So moving on to our next topic, it is P47 protein. P47 protein, the mechanical chaperone. We'll see what is the context of it. In September 2025, a landmark study by Indian researchers at the S.N. Bose National Center for Basic Sciences in Kolkata, which is an autonomous institute under the Department of Science and Technology, fundamentally changed how science understands the P47 protein. Okay. Now, what is this P47 protein of now? Let us see the historical understanding. Historically, P47 was classified simply as a cofactor or a passive helper to a larger cellular mechanism, larger cellular machine called the P97 complex. Called the P97 complex. Its known roles were limited to assisting in the protein trafficking, membrane fusion and breaking down waste proteins. These are few functions of this P47 combined with the P97 complex. Okay. Next, let us see what is the new discovery now. The new discovery is using single molecular magnetic tweezers. To mimic the cellular stress. To mimic the cellular stress inside the cell, researchers discovered the P47 is actually an autonomous, highly active mechanical chaperone. Next, what is mechanism in action? How it works? Protein inside the living cells constantly faces extreme mechanical stress and pulling forces. Okay, the proteins inside the cell, they keeps moving on due to all the functions. These proteins experiences extreme mechanical stress and pulling forces, like being dragged through narrow cellular pores from the endoplasmic reticulum to the cytoplasm. Endoplasmic reticulum, everybody would be knowing about this, right? It will be in this type of structure with ribosomes on it. Okay? We have rough endoplasmic reticulum and smooth endoplasmic reticulum in the cell. Okay, so like being dragged through the narrow cellular pores from the endoplasmic reticulum to the cytoplasm, these proteins inside the living cells will be disturbed completely. So this stress can be caused them to misfold or break. Sometimes due to this stress, these proteins may misfold or they can break also, leading to cell death or disease. So now the chaperone role. What is the chaperone role does is, in P47 acts as a protective shield. In this protein, this chaperone acts as a protective shield. It binds to the mechanically stretched polypeptides. Generally the unfolded proteins, and stabilizes them under immense stress. They stabilize these proteins under immense stress so that doesn't break or leads to death. Now, refolding. How this refolding happens? It prevents them from tangling or misfolding. Tangling means coming in collision with one another and tangling to each other. And misfolding during transport, and actively guides them to refold correctly. Once the mechanical stress is removed. Okay, this is how the chaperone acts in this P47 protein. Next, let's see the medical significance of this and few applications also. What is the medical significance? This discovery opens up entirely new avenues for precision medical aimed at targeting diseases caused by protein instability under physical stress. Okay, caused by protein instability under the physical stress. Few diseases like cardiomyopathy. Cardiomyopathy, what it does? It is a disease of the heart muscles. Where proteins are constantly under the mechanical stress of beating heart. Okay, this is called cardiomyoptosis. Next, coming to laminoparesis. Laminoparesis. Laminoparesis, it is a genetic disorder which is linked to instability of proteins in cell nucleus. Okay. Now, example like progeria, which causes the rapid aging. These are few medical significance and applications of P47 protein. Okay. So this is our today topics. I hope everyone liked it and helpful for you all. So to get a detailed content or complete notes of this content, do visit our website and download it from there. Do like and subscribe our YouTube channel for your UPSC preparation, and do follow our Telegram channel and Instagram channel as well. All the links are provided in the description below. Please do check it out, and I'll see you tomorrow with another session of Science and Technology and Environment in the Mission Surya Phase 3 of STEM Revision Program. Thank you.